All right, it's my pleasure to introduce Jamison. He's originally from Chicago, Illinois. He finished his Bachelor of Science in Ecology for Environmental Science at the University of North Texas. He is currently a master's student under the co-advisorship of Dr. Timothy Bruce and Dr. Ian Butts in the Fish Health and Fish Reproduction Labs at Auburn University. Jamison's thesis work is investigating the interactions between nutrition, health, and disease susceptibility surrounding partial replacement of fish meal with soy-based proteins in cultured Florida largemouth bass. So Jamison, you can take it away.
Awesome. Thank you, Doctor. Thank you for the great introduction. Good afternoon or good morning, everyone. My name is Jamison Semla. I'm a master's student under Dr. Timothy Bruce's Fish Health Lab here at Auburn University. And today I would like to present some of my work under my master's thesis, which is the incorporation and evaluation of soy ingredients in juvenile Florida largemouth bass. To begin, I would like to provide a brief introduction on the largemouth bass. For years now, the largemouth bass has been considered a trophy fish to many anglers on a global scale, but in recent time has built a reputation of becoming one of the most valuable freshwater cultured species in the industry. Due to increased demand and attention brought in by sport fishing tournaments, after-school extracurricular activities, and additional attention brought in by the fish has brought in an increased demand and production as a food fish and a sport fish. And just in the United States alone, the largemouth bass sports market is roughly about $60 billion and will continue to increase over time. This species is a favorite among culturists due to its ability to be easily caught, short rearing cycles, rapid growth rates, exquisite and rich meats, and its overall alluring appearances. Just to provide a brief introduction on aquafeed, currently fish meal is the choice source of protein in formulated diets in the aquaculture industry due to its high levels of digestibility, variety of amino acids, and its consistent palatability. But due to the influx of prices and availability of fish meal, the demand for it cannot keep up with the industry as it continues to grow. So it is dire for the industry to look into and investigate alternative sources of protein, both from a economic and a environmental point of view. One aspect or one alley that investigation is currently being conducted on would be with soybean meal. And the choice reason why soybean meal is a good alternative is due to its high levels of protein, supplements of amino acid, and cheap culture production. To go down and break down the investigation of our trial here, I provided a overall primary objective for this study, which would be evaluation of the incorporation of soy protein ingredients in diet formulations for juvenile largemouth bass fingerlings. And under the umbrella of that, we have three sub-objectives, which would be to determine the growth performance in largemouth bass, characterize any gastrointestinal health due to soybean meal, and overall determine the susceptibility to Colliner's disease. And with that, the hypothesis based on previous literature, we suspect or hypothesize that there would be no effect on the growth performance of the largemouth bass, but with only mild adverse effects on the health. Diving into the methods and material of this study, it was an integrative process with several sub-objectives to hit the target primary objective. And for what we did is what we started out was formulated 4 feeds, 3 of which was a partial replacement, more of a uniformity of roughly about 30% replacement with soybean meal. And we conducted a 14-week study on the fingerlings on average about 6 grams to begin with, and 16 recirculate aquaculture tanks with 4 tanks per diet. And we stocked each tank with 50 largemouth bass per tank. And at the conclusion of the growth performance study, we sampled 6 largemouth bass per tank for whole blood for lysozyme assay. We also sampled segments of the distal intestine for gene expression and histology evaluation. And we also collected samples of the fecal. And if the fecal was not accessible in PASS at the time, we took a small sliver of the distal intestine to use in lieu for a microbiota assessment. And finally to this, we wanted to look at the susceptibility to Colliner's disease. So what we set up was an immersion challenge in total of 16 flow-through isolated tanks with 20 largemouth bass per tank. And with that, we had 3 infected replicate tanks per diet and 1 mock tank per diet. And in this slide, I just wanted to highlight just the ingredient composition of each diet that I had. And the highlights would be, for this slide, would be the protein sources and what we did replacement with. And moving over to the results now of my presentation and my research, we are looking at the growth performance. Samples for growth metrics were collected at the initial, every 2 weeks, and at the conclusion of the trial. And 6 different growth metric indices were assessed with using a 2-way repeated measures of NOVA. And what we can see from each growth metric is that there was no significant difference over time through the treatment, but there was no significant difference between the interactions between the treatment and time, but there was a significant difference in time due to as the fish were continuing to grow throughout this period. Moving over to the second objective, whole blood was sampled from the fish at the conclusion of the 14-week study. The whole blood was centrifuged down to separate the layer of the remaining composition of the whole blood and the sura. The sura was subjected to a lysozyme activity assay. And from the model above, we can see that there was a significant difference in the lysozyme activity level and all 4 diets, with the soy protein concentrate just having slightly more of abundant of activity. Moving over to the microbe abundance or the microbiology part of this study, the fecal samples and intestine samples were collected and sent to an external laboratory for a 16S amplification of the V3, V4 region. And then these results were sent back to us and were analyzed in-house with the Chime pipeline and with the use of our studio. And above is a model representing the most abundant microbes found in the feed. And what we can see, the most abundant bacteria that was found was the Fusiobacterium, the Geobacillus, the Peptostreptococcus, and the Photobacterium. And then moving over to the fecal samples and intestines to look at more of overview of the GI tract of the fish following the growth performance study. We can see in the models above, I represented both the family and at the general level of the microbes that were present. And what we can see was a high abundance of Cetobacterium, Mycoplasma, Phylobacterium, and also another interesting observation that we did see in this study is that in all replicates of my tanks, but it was more abundant in the soybean meal fish, was a presence of a amphibian-based chlamydia. At this time, we don't know if it was anything pathogenic or just living inside the intestinal tract, along with the other microbes living in cohesion. To further this investigation on the microbiota assessment, we subjected all the results to a alpha diversity. And what we've seen based on the species richness, the species evenness, and the Shannon's diversity, but by treatment of all 4 of the treatments, we've seen that there was no significant difference. Moving forward, we also looked at a beta diversity assessment to look at the interaction between treatments through a unweighted and weighted unifract. And what we could see above in the unweighted unifract that there was no significant difference in presence and absence, but looking at the weighted, we did see a significant difference, and that was due to a slight difference between the soybean meal and the soy protein concentrate groups. Moving over to the histology evaluation, to first start, we looked at the qualitative based on grading scoring of 5 independent blind reviewers. And we were looking at the qualitative point, and what we've seen from the lamina propria, the connective tissue, and presence of vascularization between all 4 diets, we did not see a significant difference in the intestinal system. But on the flip side, when we looked at the quantitative histology with measuring different aspects of the distal intestinal tract, we did see several significant differences, which included the height and thickness of the mucosal fold, the subepithelial layer of the mucosal fold, and the thickness of the lamina propria. And what we can observe is that the soybean meal diet group had the highest and thickest mucosal fold, the thickest subepithelial layer, and the thickest lamina propria. And what we suspect that this diet group showed that at least some mild inflammation caused by soybean meal in comparison to the other. And the final process of the second objective of the study, we looked at gene expression of the distal intestines. And we evaluated 4 different inflammatory cytokine proteins for this study. And what we found from each of them for all the diets is that there was not a significant difference in inflammatory presentation in the distal intestine of the bass. And to the final objective of this study, what we were looking at is a immersion challenge with a low dose of Flabobacterium colinaire strain ARS-LNB-23-5, which was provided by Dr. LaFrance and the USDA ARS station here at Auburn University that was isolated from the bass from the previous year. And what we can observe from the model above in the Kaplan-Meier survivability is that a majority of the mortalities did occur around hour 12. But when we looked at it from a statistical point of view, we can see among the 3 different soybean-based diet fish and the control is that there was a significant difference in the survivability of the fish for this emergent challenge. And what we can see is that the soy protein concentrate did slightly have a higher survivability than the rest of the groups. So in conclusion of my study, and the highlight of this trial, what we're trying to target is that there was no significant differences in the growth performance of the largemouth bass when they were fed a formulated experimental diet with a partial replacement of soybean meal of each of the 3 variants. There was no differences in inflammatory regulations of the cytokine proteins, but there was some small differences in the histology, serocomposition, and gut microbiology, and significant differences in the survivability of the immersion challenge. Moving forward with future directions of research of largemouth bass culture with soy-based feeds, and it's things that I'm going to be looking into the remainder of my master's here at Auburn University, would be looking at threshold limit replacements of soybean for largemouth bass. And for that, we'll be using a gradient replacement. The next thing would be looking at a soy-based protein with a reduction in anti-nutritional factors. And for an example for that would be the low oligosaccharides. And the next, we would look at the incorporation of probiotics with soy-based proteins. And another step would be pushing it from inside from a recirculated system to outdoor systems, looking at the culturing of largemouth bass when fed soy-based proteins in either aquaponic settings or raceway settings. And another aspect or alley that could be looked at for the further investigation of largemouth bass in the industry, especially with the soy-based feeds incorporated into the study, would be the susceptibility to other high-risk pathogens or polyinfections. And this would include Aeromonas or red sores disease or largemouth bass virus. And with that, I would like to thank you all for listening to my presentation. I would like to thank my funders, the Soy Aquaculture Alliance, for funding this project. I would like to thank Auburn University, the School of Fisheries, Aquaculture, and Aquatic Sciences for bringing me on this project and allowing me to conduct this research. I would like to thank all my peers at the EW Shell who has helped along with this project. This would also include my lab mates and my lab manager under the Bruce Fish Health Laboratory. I would also finally like to thank my committee for the countless hours that they put in with the study and will continue to do so. And here's my references that were incorporated into this study. And with that, I would like to open up the discussion for any questions.
Great job, Jamison.
Thank you,
Folks,
sir.
if you have questions, feel free to drop them in the chat or unmute yourself. We have about 2 minutes. All right, Jamison, you must have filled all the gaps. Thanks again for the presentation.
Of course, no problem. If anybody does have any further questions, even after this seminar, I will drop my email into the chat and we can further reach out and get back to you between myself and my team on any questions anybody would have.
Great.